Basics of Medical Biochemistry. First edition, 2022. Chapter 7. Bioenergetics. Part 1 of 2. By Professor Dr. Muhammad Jawad. MBBS, Tau, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 7. Bioenergetics. 7.1 Introduction. Bioenergetics is the quantitative study of energy transductions in living cells. It is the branch of biochemistry concerned with the energy involved in making and breaking chemical bonds in the molecules found in biological organisms. All living organisms have the ability to produce energy and channel it into biological work. They obtain energy from organic and inorganic resources. For example, autotrophs can produce ADP by photosynthesis. Heterotrophs, including humans, consume organic compounds like carbohydrates, fats, and proteins to get energy. The amount of energy obtained from these compounds is lower than the amount present in the food. It is due to some losses during digestion, metabolism, and thermogenesis. Bioenergetics is concerned to see how living organisms carry out energy transductions and conversions of one form of energy to another form. Basic concepts. The role of energy is fundamental to all biological processes such as growth, development, reproduction, muscle contraction, nerve conduction, and metabolism. The thermodynamics in living organisms is called bioenergetics. Energy is the capacity of a body to perform work. Kinetic energy or energy of motion is the form of energy exhibited by doing work or movement like heat, light, etc. Potential energy is the energy content of a matter, because of its arrangement or position, like the chemical energy of a gas or food, water behind a dam, etc. Higher organisms use the chemical energy in fuels, carbohydrates, lipids, to bring about the synthesis of complex macromolecules from simple precursors. Higher organisms can convert the chemical energy into concentration gradients and electrical gradients, into motion and heat, and in a few organisms into light, fireflies, some deep-sea fishes. Biological energy transductions follow the laws of thermodynamics. Adenosine triphosphate, ADP, is the normal molecule that cells use to store and release energy. ADP provides energy usually through phosphoryl group transfer. A protein could be such an acceptor. The food materials are generally combined with oxygen during oxidation to discharge energy. However, some organisms can oxidize their food chemicals anaerobically to get the required energy. The food materials are oxidized at a slow pace, sufficient enough to keep organisms well protected from the actual production of fire. Thus, the utilization of these materials is an act of slow combustion. The energy of food is released slowly and captured in the bonds of ADP. 7.2 Laws of Thermodynamics and Energy Changes. These laws describe the particulars of the transport of heat in thermodynamic processes. 7.2.10 Law of Thermodynamics. It states that if two bodies are each in thermal equilibrium with some third body, then they are also in equilibrium with each other. It means that the amount of energy in the universe is constant. 7.2.2 First Law of Thermodynamics. Energy can neither be created nor destroyed. It can only change from one form to another. 7.2.3 Second Law of Thermodynamics. It states that there is a natural tendency of any isolated system to degenerate into a more disordered state, i.e., to increase the entropy. For example, broken glass has less order, more entropy, than intact one, and it is more difficult to repair broken glass, reducing its entropy, than to break an intact one, increasing its entropy. The universe always tends toward increasing disorder. In all natural processes, the entropy of the universe increases. 7.2.4 Third Law of Thermodynamics. It is about the entropy of an object at absolute zero temperature, and implies that it is impossible to cool a system all the way to exactly absolute zero. As temperature approaches absolute zero, the entropy of a system approaches a constant minimum. At absolute zero of temperature, 0 K equals 273.15 degrees C equals 459.67 degree F, all atoms of the system stop moving, K equals Kelvin, which means absolute temperature. 7.2.5 Entropy, S. It is the amount of disorder in a system. In most but not all cases, it is heat. S changes in entropy, plus S means an increase in randomness and S means a decrease in randomness. For example, the melting of ice to a liquid state of water is the gain or increase in entropy. Because there is more molecular disorder in fluid than in solid state. For most spontaneous reactions the change of entropy is plus S. The unit of S is joules or cal/mol.K. 7.2.6 Enthalpy, H. It is the total heat of a system, heat content of reactant and product. Change in enthalpy of a reaction at constant temperature and pressure is denoted by H. It may be positive, for endergonic reactions, and negative, for exergonic reactions. Enthalpy reflects the number and kinds of chemical bonds in the reactants and products. The unit of H is kcal/mol or kj/mol. Generally, reactions occurring spontaneously is exergonic. But it is not the rule, that's why the sign and value of H cannot give any indication about the direction of the reaction. 7.2.7 Free energy, G. It is the amount of usable energy in a system that can be used to perform work. It is also known as Gibbs free energy. Ig equals free energy difference of a system in any condition. 2G0 equals free energy difference of a system in standard condition, at 25 degrees Celsius and 1 atmosphere pressure while the concentration of reactants and products are 1 mol/L. 3G0 equals free energy difference of a system in standard condition at pH 7 i.e., H plus is 10 to 7 mol/L. Negative G, G is negative for exergonic reactions, release of free energy. It is to be noted that most spontaneous reactions are exergonic in nature, so they have G. Positive G, G is positive for endergonic, absorption of energy, reactions. 0 G, when the reaction is in equilibrium, G equals 0. The magnitude of free energy is the same for forward and reverse reactions but with opposite signs. Plus, for back reaction and minus for forward reaction. Living organisms preserve their internal order by taking free energy from their surroundings in the form of nutrients or sunlight and returning to their surroundings an equal amount of energy as heat and entropy. The unit of G is kcal/mol or kj/mol. 7.2.8 Relationship between enthalpy, H, entropy, S, and free energy, G. G equals H, Ts, T equals absolute temperature. Entropy is maximum when the reaction is at equilibrium. Free energy is the magnitude of the force driving the reaction toward the equilibrium. Free energy becomes maximum when the reaction attains equilibrium. Calorie, cal. It is the unit of heat. 1 kilocalorie, kcal, equals 1000 calories. 1 kilocalorie equals 4.2 kilojoule, kj. 7.2.9 Significance of G and G0. G0 is the difference between the free energy content of the products and the free energy contents of the reactants under standard conditions. G0 equals G0 products, G0 reactants. When G0 is negative, the products contain less free energy than the reactants and the reaction will proceed spontaneously under standard conditions. When G0 is positive, the products contain more free energy than the reactants and the reaction will tend to go in the reverse direction under standard conditions. Thus, G0 tells us in which direction and how far a given reaction must go to reach equilibrium when the initial concentration of each component is 1 mol/L, 1 m or 1 molar, the pH is 7 and the temperature is 25 degrees Celsius. Delta G of any reaction proceeding spontaneously toward its equilibrium is always negative, becomes less negative as the reaction proceeds, and it becomes 0 at the point of equilibrium, indicating that no more work can be done by the reaction. Consider a general reaction A plus B C plus D at constant temperature and pressure. It represents the following relationship: CD AB, where R equals gas constant, 1.987 cal/mol. K, T equals absolute temperature, K, Ln equals natural logarithm, and A, B, C and D are the actual concentrations. The standard values of G0, R and T are fixed. At the start of the reaction, C and D are very small, but as the reaction progressed their concentrations increased gradually till the equilibrium. Similarly, G becomes less negative and eventually becomes zero at equilibrium. At the point of equilibrium, the concentrations of A, B and C, D are equal and the rate of forward and back reaction is the same. When the reaction is at equilibrium, there is no force driving the reaction in either direction and delta G is zero. The above equation at equilibrium can be represented in the following manner: C eq D eq, A eq B eq, or C eq D eq, A eq B eq. The above equation is expressing the relationship between G0 and the equilibrium constant Keq. It is concluded that G0 is negative when Keq is more than 1, G0 is 0 when Keq is 1 and G0 is positive when Keq is less than 1. The relationship between G0 and Keq is helpful in predicting the direction of the reaction. Reaction is at equilibrium when Keq equals 1 and G0 equals 0. Forward reaction when Keq greater than 1 and G0 0, negative. Back reaction when Keq 1 and G0 greater than 0, positive. However, a reaction can move in the forward direction with positive G0 when G is negative. It happens when the ratio of products to reactants, products/reactants, is very small, 1. This situation occurred in many metabolic pathways when products are being constantly removed. Standard free energy changes, G0, are additive, in the case of two sequential chemical reactions as given below. ABG01 and BCG02. Since the two reactions are sequential, they can be represented as ACG0 total. The G0 values of sequential reactions are additive, i.e. G0 total equals G01 plus G02. This principle of bioenergetics explains how a thermodynamically unfavorable, endergonic, reaction can be driven in the forward direction by coupling it to a highly exergonic reaction through a common intermediate. All endergonic reactions are coupled to an exergonic reaction. There is an energy cycle in cells that links anabolic and catabolic reactions. For example, consider the first step of glycolysis. Glucose plus pi glucose 6-phosphate plus H2O, G0 equals 13.8 kJ/mol. As G0 greater than 0, the reaction is not spontaneous. Another very exergonic accompanying cellular reaction is the hydrolysis of ADP. ADP plus H2O ADP plus pi, G0 equals 30.5 kJ/mol. These two reactions share the common intermediates H2O and pi and may be expressed as sequential reactions. Glucose plus pi glucose 6-phosphate plus H2O. ADP plus H2O ADP plus pi. Glucose plus ADP glucose 6-phosphate plus ADP. The overall standard free energy changes, G0 equals 13.8 kJ/mol plus 30.5 kJ/mol, equals 16.7 kJ/mol, and overall reaction is exergonic. Energy stored in ADP is used to drive to the synthesis of glucose 6-phosphate, even though its formation from glucose and pi is endergonic. This strategy is feasible only if required compounds like ADP are continuously available. Table 7.1 G0 values of some important biochemical reactions. Biochemical reaction. G0, kcal/mol. Fructose 6-phosphate plus H2O fructose plus pi. 3.8. ADP plus H2O ADP plus pi. 7.3. ADP plus H2O AMP plus PPI. 7.7. Creatine phosphate plus H2O creatine plus pi. 10.3 Glucose 6-phosphate plus H2O glucose plus pi. 3.3 7.3 Types of reactions. Thermodynamically, the reactions may be heat-absorbing or heat-releasing. 1. Endergonic/endothermic reactions. In these reactions energy is absorbed, they have plus G greater than 0. These reactions are not spontaneous. 2. Exergonic/exothermic reactions. In these reactions energy is released, they have G0. These reactions are spontaneous. At the equilibrium state of the reaction, G equals 0, i.e., heat loss and absorption are equal. The total of the chemical activities of all cells is called cellular metabolism. Anabolic pathways are endergonic reactions. They consume energy to build complicated molecules, protein, glycogen, and lipid, from simpler compounds. Catabolic pathways are exergonic reactions. They release energy by breaking down complex molecules into simpler compounds such as glycolysis and glycogenolysis. 7.3.1 Oxidation. It is the chemical process represented by any of the following changes: gain of oxygen, loss of hydrogen, loss of electron, increase in oxidation state. 7.3.2 Reduction. It is the chemical process represented by any of the following changes: loss of oxygen, gain of hydrogen, gain of electron, decrease in oxidation state. Figure 7.1 Characteristic features of endergonic and exergonic reactions. 7.3.3 Redox reaction. A redox or oxidation-reduction reaction is a type of chemical reaction that involves a transfer of electrons between two species. Oxidation and reduction always occur simultaneously. Oxidizing and reducing agents. An oxidizing agent or oxidant is a substance that oxidizes something else, itself reduced. A reducing agent or reductant reduces something else, itself oxidized. Oxidation and reduction in terms of oxygen transfer. Oxidation is the gain of oxygen and reduction is the loss of oxygen. For example, in the extraction of iron from its ore. Figure 7.2 Oxidation and reduction in terms of oxygen transfer. In the above example, iron 3 oxide is the oxidizing agent. It oxidizes carbon monoxide to carbon dioxide but itself reduced to iron. Similarly, carbon monoxide is the reducing agent. It reduces iron 3 oxide to iron but itself oxidized to carbon dioxide. Oxidation and reduction in terms of hydrogen transfer. Oxidation is the loss of hydrogen and reduction is the gain of hydrogen. For example, the reaction between lactate and NAD+. Lactate is oxidized to pyruvate and NAD+ is reduced to NADH plus H+. Figure 7.3 Oxidation and reduction in terms of hydrogen transfer. Oxidation and reduction in terms of electron transfer. Oxidation is the loss of electrons and reduction is the gain of electrons. For example, in a reaction between copper 2 oxide and magnesium, they are reduced and oxidized respectively. Figure 7.4 Oxidation and reduction in terms of electron transfer. 7.4 Reduction oxidation, redox, potentials. Redox reactions involve the transfer of electrons from one chemical species to another. All oxidation-reduction, redox, reactions can be divided into two half reactions, one in which a chemical species undergoes oxidation and one in which another chemical species undergoes reduction. In the reduction part of the reaction, the driving force is the reduction potential. Similarly, in the oxidation part of the reaction, the driving force is the oxidation potential. So, the redox potential is the reduction/oxidation potential of a compound measured under standard conditions against a standard reference half cell. 7.4.1 Redox pairs. In redox reactions, one substance is oxidized while the other is reduced. They form a pair called a redox pair. For example, NAD+/NADH plus H+, FAD/FADH2, FMN/FMNH2, etc. The oxidized plus the reduced form of each chemical species is referred to as an electrochemical half cell. Two half cells having at least one common intermediate comprise a complete, coupled, redox reaction. Coupled electrochemical half cells have the thermodynamic properties of other coupled chemical reactions. If one half cell is far from electrochemical equilibrium, its tendency to achieve equilibrium, i.e., to gain or lose electrons, can be used to alter the equilibrium position of a coupled half cell. 7.4.2 Redox potential. It is the measure of the tendency of oxidant to gain electrons, to become reduced. It is a potential energy. In other words, it is the ability of a substance to get electrons and get reduced itself. In a redox reactions, electrons move from compounds with lower reduction potential, more negative, to compounds with higher reduction potential, more positive. Reductant oxidant plus E. Oxidant plus E reductant. The redox potential is symbolized by E and measured in volts, V. E equals redox potential of a system in any condition. E0 equals redox potential of a system in standard condition, 25 OC and 1 atmosphere pressure. E0 equals redox potential of a system in the standard condition in a biological system at pH 7.0. The amount of energy released from an oxidation-reduction reaction can be predicted by the following relationship, Nernst equation. G0 equals NFE0, where N is the number of transferred electrons, 1, 2, 3, F is the Faraday constant that is 23.06 kcal/volt/mole or 94.4 kJ/volt/mole. E is measured in volts and G is measured in kcal/mole or kJ/mole. In other words, energy, work, can be derived from the transfer of electrons as the oxidation of foods can be used to synthesize ADP. For the oxidation of NADH, the standard biological reduction potential is 52.6 kcal/mole. With a free energy change of 52.6 kcal/mole, it is clear that NADH oxidation has the potential for driving the synthesis of many ATPs since the standard free energy for the reaction below is plus 7.3 kcal/mole. ADP plus pi ADP. Classically, the explanation of ADP synthesis through oxidation of reduced electron carriers indicated that 3 moles of ADP could be generated for every mole of NADH and 2 moles for every mole of FADH2. However, direct chemical analysis has shown that for every 2 electrons transferred from NADH to oxygen, 2.5 equivalents of ADP are synthesized and 1.5 for FADH2. Table 7.2 Standard redox potentials, E0, of some biologically important compounds. Oxidant. Reductant. N, number of electrons transferred. E0, volts. NAD plus. NADH. 2. 0.32. Acetaldehyde. Ethanol. 2. 0.20. Pyruvate. Lactate. 2. 0.19. Oxalostate. Malate. 2. 0.17. 1/2O2 plus 2H plus. H2O. 2. Plus 0.82. Oxidant can oxidize any compound which has a redox potential less than its. Reductant can reduce any compound which has redox potential more than its redox potential. 7.5 Compounds of high energy bonds. Living organisms acquire free energy from nutrient molecules. Cells transform this free energy into ADP and other energy-rich compounds. These compounds are capable of providing energy for biological work at a constant temperature. Compounds with high energy bonds are said to have high group transfer potential. For example, pi may be spontaneously cleaved from ADP for transfer to another compound, example to a hydroxyl group on glucose or to protein. 7.5.1 High energy phosphate compounds. These include ADP, GTP, CTP, UTP, etc. They can give energy in the range of 5-12 kcal/mole. Figure 7.5 Structure of ADP, ADP, AMP, and adenosine. Phosphoanhydride bonds. formed by splitting out H2O between two phosphoric acids or between carboxylic and phosphoric acids, have a large negative G of hydrolysis. Phosphoanhydride linkages are said to be high-energy bonds. High-energy bonds are represented by the symbol P. Represents a phosphate group with a large negative G of hydrolysis. Functions of ADP: it is involved in muscle contraction, sodium-potassium pump, maintaining resting membrane potential, reabsorption of Na+ in proximal convoluted tubules, synthesis and secretion of various substances, as well as a detoxification process. Release of energy from ADP. ADP plus H2O ADP plus pi, Go equals 31 kJ/mol or 7.3 kcal/mol. ADP plus H2O amp plus PPI, pyrophosphate, Go equals 32.3 kJ/mol or 7.7 kcal/mol. During periods of intense demand for ADP, the cell lowers the ADP concentration and at the same time acquires ADP, by the action of adenylate kinase in the presence of Mg2+ ions. 2-ADP ADP plus AMP. This reaction is fully reversible, so after the end of the intense demand for ADP, the enzyme can recycle AMP by converting it to ADP, which can then be phosphorylated to ADP in mitochondria. 7.5.2 Creatine phosphate. Phosphocreatine. Creatine phosphate is another high-energy phosphate bond-containing compound. It is present in muscles as a stored high-energy phosphate compound. Its hydrolysis can produce energy up to 10 kcal/mol. Formation of creatine phosphate. Its formation takes place at rest. Creatine plus ADP. Creatine phosphate plus ADP, by enzyme creatine kinase. Breakdown of creatine phosphate. Breakdown of creatine phosphate takes place during muscle contraction, when all ATPs have been used up, to replenish ADP. Creatine phosphate plus ADP. Creatine plus ADP, by enzyme creatine kinase. G0 equals 42 kJ/mol or 10 kcal/mol. Some ordinary energy phosphate compounds include glucose 6-phosphate and glycerol 3-phosphate. Their energy-giving capacity is 4 kcal/mol. Phosphenol pyruvate, 14.8 kcal/mol, and 1,3 bisphosphate, 11.8 kcal/mol, are also high-energy phosphate compounds. 7.5.3 High-energy thioesters. Thioesters are organic compounds having general formula, R-CoSr. On hydrolysis, they yield carboxylic acid and mercaptan, HSR. Acetyl-CoA, CH3-CoS-CoA, is an example of high-energy thioesters. Its G0 equals 31.5 kJ/mol or 7.5 kcal/mol. Acetyl-CoA is produced from pyruvic acid and beta-oxidation of fatty acids. It is oxidized in the TCA cycle. Functions: besides energy generation, it is also involved in the biosynthesis of fatty acids, cholesterol, acetylcholine, acetylated sugars, acetyl peptides, and many detoxification reactions. Role of high-energy phosphates in energy capture and transfer. The term high-energy phosphate bond used to describe the PO bond broken in hydrolysis reactions is incorrect and misleading, as it wrongly suggests that the bond itself contains the energy. The breaking of all chemical bonds requires an input of energy. The free energy released by the hydrolysis of phosphate compounds does not come from the specific bond that is broken. In fact, it results from the products of the reaction having a lower free energy content than the reactants. As it is evident from the additivity of free energy changes in many sequential reactions. For example, cleavage of pi from phosphenol pyruvate, PEP, releases more energy than is needed to drive to condensation of pi with ADP. The direct donation of a phosphoryl group from PEP to ADP is thermodynamically feasible. Phosphenol pyruvate, PEP, plus H2O pyruvate plus pi, G0 equals 61.9 kJ/mol. ADP plus pi ADP plus H2O, G0 equals plus 30.5 kJ/mol. Phosphenol pyruvate, PEP, plus ADP pyruvate plus ADP, G0 equals 31.4 kJ/mol. The overall reaction does not involve pi. The PEP donates a phosphoryl group directly to ADP. Based on standard free energy changes of hydrolysis, the phosphorylated compounds can be described as having a high or low phosphoryl group transfer potential. Most catabolism is directed toward the synthesis of high-energy phosphate compounds, but their formation is not an end in itself, they are the means to activate a wide variety of compounds for further chemical transformation. The transfer of a phosphoryl group to a compound effectively puts free energy into that compound, so that it has more free energy to give up during subsequent metabolic transformations. ADP can carry energy from high-energy phosphate compounds produced by catabolism to compounds such as glucose, converting them into more reactive species. ADP provided a chemical link between catabolism and anabolism. The exergonic conversion of ADP is coupled to many endergonic processes in living organisms. Thus, ADP serves as the universal energy currency in all living cells. 7.6 Enzymes and coenzymes for oxidation and reduction reactions. Oxidoreductases, these enzymes catalyze various oxidation reduction, redox, reactions. They include oxidases, dehydrogenases, hydroperoxidases, and oxygenases. Coenzymes. Main examples of coenzymes involved in redox reactions are NAD+, NADP+, FAD, FMN, coenzyme Q, Pfau proteins, etc. Coenzymes worked as hydrogen acceptors. They exist in oxidized, NAD+, and reduced, NADH+, H+, forms. These are non-protein parts of respective enzymes. All coenzymes are involved in redox reactions for energy generation except NADP+. The NADP+ is produced in the HMP shunt and utilized in the formation of reduced glutathione, fatty acids, cholesterol, etc. Reduced coenzymes are oxidized by a complex series of reactions by etc. present in the cytochromes of mitochondria. The end product of their oxidation is water and heat, enough to produce ADP. As electrons in a CO bond are associated more with O, increased oxidation of a C atom means an increased number of CO bonds. The oxidation of carbon is spontaneous, i.e., energy yielding. Figure 7.6 Oxidation of carbon atoms, increasing from left to right. 1 Nicotinamide adenine dinucleotide, NAD+. It is derived from niacin, vitamin B3, a member of the vitamin B complex. Its functional part is nicotinamide. It acts as a coenzyme for many dehydrogenases. The nicotinamide ring, derived from niacin, accepts 2E and 1H+, a hydride, in going to the reduced state, NADH+ H+. It is mainly involved in ADP-generating reactions. Figure 7.7 Structure of nicotinamide adenine dinucleotide, NAD+, right, and NADP+, left. 2 Nicotinamide adenine dinucleotide phosphate, NADP+. Its structure is similar to NAD+, except for pi. It also exists in oxidized, NADP+, and reduced, NADH+, forms. It is mainly involved in biosynthetic pathways, example fatty acid and cholesterol synthesis. The electron transfer reaction by NAD+ may be summarized as NAD++ 2E+ H+ NADH. It may also be expressed as NAD++ 2E+ 2H+ NADH+ H+. Figure 7.8 Role of NAD+ in the transfer of electrons, riboflavin. 3 Flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN. These are derived from riboflavin, vitamin B2, a member of the vitamin B complex. They serve as E acceptors. Their functional part is dimethyl isoalloxazine ring, which undergoes reduction/oxidation. FAD accepts 2E and 2H+ to achieves reduced state FADH2, note, H atom equals H++ E. FAD plus 2E plus 2H plus FADH2. Figure 7.9 Structure of flavin adenine dinucleotide, FAD, and flavin mononucleotide, FMN. 4 Coenzyme Q, ubiquinone. It is a vitamin-like fat-soluble substance found in the mitochondria. It is actively involved in the electron transport chain and generation of ADP via the oxidative phosphorylation process. Its hydrophobic tail is composed of 6 to 10 5-carbon isoprenoid units. It is highly hydrophobic and lipophilic. It diffuses freely in the lipid bilayer of the inner mitochondrial membrane. The coenzyme Q with 10 isoprenoid units is termed coenzyme Q10. It can accept 1 or 2 H atoms from FADH2 and FMNH2. 5 iron-sulfur, Pfas, proteins. Many non-heme iron-sulfur complex proteins are found in mitochondria. Iron is bound to sulfur either in the elemental form or to the thiol in the cysteine side chain. These proteins have a significant role in the oxidation-reduction reactions of electron transport. Both complex I and complex II of oxidative phosphorylation have multiple Pfas clusters. Some Pfas proteins are also involved in the regulation of gene expression and DNA repair. Figure 7.10 Structure of coenzyme Q10, ubiquinol, reduced form, ubizemikinin, intermediate form, and ubiquinone, oxidized form. 7.7 Role of mitochondria in bioenergetics. The mitochondria are specialized rod or oval-shaped cellular organelles working as the powerhouse of the cell. Mitochondria are present in virtually every cell of the body. Mitochondria have two membranes, an outer and an inner membrane. The outer membrane is permeable to many small molecules and ions because it contains large protein channels called porins. The inner membrane is impermeable to most ions and polar molecules. The space between the two membranes is known as the intermembrane space. Inside the inner membrane is a space known as the matrix. The mitochondrial matrix contains the enzymes required for the TCA cycle, oxidative phosphorylation, and the oxidation of fatty acids. The inner membrane is the site of oxidative phosphorylation. The matrix side of the inner membrane has a negative electrical charge relative to the intermembrane space due to an H+ gradient set up by the redox reaction. This charge difference is used to provide free energy, G, for the phosphorylation reaction. The intermembrane space side of the inner membrane is referred to as the P face, P equals positive. The matrix side of the inner membrane is referred to as the N face, N equals negative. The complexes of electron transport chains are embedded in the inner mitochondrial membrane. Although the inner membrane is mostly impermeable, it contains special H+, proton, channels and pumps that enable the coupling of the redox reaction involving NADH plus H plus and O2 to the phosphorylation reaction of ADP. Mitochondria and apoptosis. Mitochondria play key roles in activating apoptosis in mammalian cells. Apoptosis is mediated by an intrinsic pathway in mitochondria causing the formation of pores in the outer mitochondrial membrane. The cytochrome C leaves the intermembrane space and enters to the cytosol, where in combination with pro-apoptotic factors it activates the group of proteolytic enzymes called caspases. Caspases are a family of cysteine proteases that serve as primary effectors during apoptosis to proteolytically dismantle most cellular structures, including the cytoskeleton, cell junctions, mitochondria, endoplasmic reticulum, Golgi apparatus, and the nucleus. Clinical aspects: a group of diseases abbreviated as MELAs, mitochondrial encephalopathy, lactic acidosis, stroke, occurs due to complex I deficiency. It is characterized by the deficient synthesis of ADP. Labor hereditary optic neuropathy, LHON, is an inherited form of vision loss in early adulthood. It is due to mutations in mitochondrial DNA. Another mitochondrial disorder is myoclonic epilepsy and ragged red fiber, MERF, disease, affecting many systems of the body, particularly the muscles and nervous system. Mitochondrial DNA mutations may also cause Alzheimer's disease, Parkinson disease, diabetes mellitus and hypertrophic cardiomyopathy. Figure 7.11 Mitochondria showing the site of the electron transport chain and oxidative phosphorylation, source, Quizlet.com. 7.8 Electron transport chain, etc., or respiratory chain. It is a group or chain of mitochondrial catalysts, enzymes and coenzymes with cytochromes, involved in the transport of reducing equivalents to molecular oxygen. The process of oxidative phosphorylation is carried out by the electron transport chain. The mitochondrial electron transport chain removes electrons from an electron donor, NADH plus H plus or coenzyme QH2, and passes them to a terminal electron acceptor, O2, via a series of redox reactions. These reactions are coupled to the creation of a proton gradient across the mitochondrial inner membrane. There are 3 proton pumps, I, 3, and 4. The resulting transmembrane proton gradient is used to make ADP via ADP synthase. 7.8.1 Cytochromes. These are of the following 2 types. 1. Mitochondrial cytochromes. These are conjugated proteins that act as enzymes. They contain one atom of iron called ferric as Fe3+, oxidized, or ferrous as Fe2+, reduced, form. One cytochrome can carry one electron at a time. Important types of mitochondrial cytochromes are B, C1, C, A and A3 or A plus A3. 2. Microsomal cytochromes. These are found in microsomes. Examples are P450 or CYPs. They are involved in the oxidation of xenobiotics and several drugs. 7.8.2 Reducing equivalent and sources of electrons. The term reducing equivalent is commonly used to designate electron equivalent participating in an oxidation-reduction reaction. Most of the energy liberated during the oxidation of proteins, carbohydrates, and lipids is made available within mitochondria as reducing equivalents, i.e., NADH plus H plus and FADH2 that arise from glycolysis, located in the cytoplasm, TCA cycle, and fatty acid oxidation, located in the mitochondrial matrix. These reducing equivalents are then aerobically oxidized in etc. with the liberation of energy which is used to synthesize ADP. The formation of ADP by this mechanism is called oxidative phosphorylation. Figure 7.12 General scheme of oxidation of NADH plus H plus and formation of ADP via oxidative phosphorylation. NADH plus H plus and FADH2 are the major sources of electrons while FMNH2 and NADH plus H plus are regarded as minor sources. 1. Formation of NADH plus H plus. These are produced in many dehydrogenation reactions. For example, imalate plus NAD plus oxalostate plus NADH plus H plus, in TCA cycle. 2. Beta-hydroxy fatty acyl CoA plus NAD plus beta-keto acyl CoA plus NADH plus H plus, in beta-oxidation of fatty acid. 2. Formation of FADH2, these are also produced in many dehydrogenation reactions. For example, isoxanate plus FAD fumarate plus FADH2, in TCA cycle. 2. Palmitoyl CoA plus FAD, unsaturated palmitoyl CoA plus FADH2, in beta-oxidation of fatty acid. 3. Formation of coenzyme QH2, it is produced either from NADH plus H plus or FADH2. I from NADH plus H plus. NAD plus ADP plus pi ADP. NADH plus H plus FMN FMNH2 plus coenzyme QH2 plus FMN. 2. From FADH2. FADH2 plus coenzyme QFAD plus coenzyme QH2. 4. Oxidation of coenzyme QH2. Coenzyme QH2 coenzyme Q plus 2H plus 2E. The 2H plus produced in the above reaction enter the pool of H plus ions in the mitochondrial matrix and 2 electrons are picked by 2 molecules of the oxidized form of cytochrome B, i.e. CytB3+. Now from here, the role of cytochromes started. 5. Role of cytochromes in the electron transport chain. I. Reduction of CytB3+. 2 CytB3++ 2E2 CytB2+. 2. Oxidation of CytB2+ and reduction of CytC13+. ADP plus pi ADP. 2 CytB2+ plus 2 CytC13 plus 2 CytB3 plus 2 CytC12+. 3. Oxidation of CytC12+ and reduction of CytC3+. 2 CytC12 plus 2 CytC3 plus 2 CytC13 plus 2 CytC2+. 4. Oxidation of CytC2+ and reduction of CytA plus A33+. ADP plus pi ADP. 2 sit C2 plus 2 sit A plus a 33 plus 2 sit C3 plus 2 sit A plus a 32 plus V oxidation of sit A plus a 32 plus 2 sit A plus a 32 plus 2H plus 1 half O2 2 sit A plus a 33 plus H2O. Exercise questions. MCQ 1. Phosphenol pyruvate, PEP, plus pyruvate plus ADP, G0 equals 31.4 kJ/mol. Which of the following reactants is missing in the reaction shown above? ADP, MP, GDP, Pi, PPI. MCQ 2. During an experiment on bioenergetics, the free energy difference of a system is measured at standard conditions with pH 7. Which of the following symbols is used to indicate the measured energy in this experiment? E, G, G0, G0, H. MCQ 3. The statement if two bodies are each in thermal equilibrium with some third body, then they are also in equilibrium with each other is defining which of the following laws of thermodynamics? Zeroth law of thermodynamics. First law of thermodynamics. Second law of thermodynamics. Third law of thermodynamics. MCQ 4. Which of the following terms is used to indicate the total heat content of reactants and products in a system? Enthalpy, entropy, free energy, redox potential. MCQ 5. Which of the following components of this reaction, Cu2 plus MgCu plus Mg2 plus, is oxidized? Cu2 plus Mg, Cu, Mg2 plus. Please write answers in the comments or contact drmjot@yahoo.com. End of Part 1 of 2 of Chapter 7. Thank you.